Carmen Nussbaum, Center of Molecular Biology, Heidelberg. Combating protein misfolding in neurodegenerative diseases.
Protein misfolding is central to many neurodegenerative diseases. For example, in Parkinson's disease, next slide please, alpha-synuclein is aggregating. And next slide. And amyloid fibril formation is dependent on the conformation of a soluble monomer that is then forming a misfolded species and beta sheet-rich conformation, which is able to self-propagate and then also grow into toxic oligomers and fibrils. And next slide, the deposits of alpha-synuclein are known to form first in a very distinct area of the brain, but during disease progression, the aggregation and toxicity is spreading in a manner similar to the infectious prions. And we are working on two main questions. The first is, are there cellular factors that influence alpha-synuclein fibril formation? And specifically, are they able to reverse fibril formation? And second, are there cellular factors that inhibit the spreading of alpha-synuclein? Next slide. Molecular chaperones are known for a long time to assist in folding and prevent the misfolding of proteins and the formation of amyloids. But it was not clear if they would also be able to disassemble these highly packed fibrils. And we could recently show that such an activity exists in human cells, and it is ATPase dependent. Next slide, please. And based on a concerted action of HSP70 together with specific co-chaperones. And next slide. The complete solubilization of alpha-synuclein fibrils into monomers reduces the toxicity. And our perspectives are, we want to try if other disease-related proteins are able to be disaggregated with this network and look at the mechanism, and also we want to explore if this action can be used as a therapeutic target. Now, to address whether alpha-synuclein is spreading and which factors are involved, we're using C. elegans because this worm is transparent, and so it allows the in vivo monitoring of cell-to-cell transmission of alpha-synuclein. And so you can see that it's only expressed in the donor cells, but taken up into vesicles that are then transmitted into neighboring cells. And we are using this system to screen for modifiers, and they impact very different ways in the pathway of alpha-synuclein propagation. So, for example, the aggregate state or the transmission between cells. And with that, I want to point out that the first part is done in Bernd Bucker's lab at the ZMPH, and the C. elegans work is done in my lab. And thank you for your attention.